Welcome to the video for chapter 6.6. We're talking about microbial communities and cell differentiation. This is the last lecture for chapter 6, so chapter 6 homework is going to be due pretty soon. So let's get into this. Okay, but first let's do a review question. So how would you classify a bacterial species that grows only in the highlighted area of the culture tube? Okay, so our highlighted area is this part outlined in blue. Okay, so is this organism aerotolerant? B, a strict anaerobe? C, a strict aerobe? D, a facultative anaerobe? Or E, a microaerophile? So pause the video, look in your notes, see if you can find the answer. Hey, so the answer for this is B. We need to be able to live in no oxygen, so that means we need an anaerobe, which is going to automatically cancel out C, because that can only live up here. Okay, so remember, a facultative, facultative anaerobe, a aerotolerant anaerobe, and microaerophiles all live in this center place. So the only one that lives in a no oxygen condition all the time would be B, the strict anaerobe. Okay, so in 6.6, we are talking about microbial communities and cell differentiation. So cell differentiation is when we take a precursor cell, so something like our human stem cells, and it's going to go through different mechanisms that are going to cause it to grow and divide and create specific proteins that are going to turn it into a specialized type of cell. Okay, so like our stem cells can become lung cells, they can become skin cells, hair cells, heart cells, lung cells, liver cells. Okay, it all has to do with that process of cell differentiation. So in chapter 6.6, we are going to talk about biofilms and endospores. Okay, so we're going to talk about how biofilms develop and the role quorum sensing has in that process. We're going to talk about biofilms and infection and why they are such a problem in the medical field right now. Okay, and then we're going to talk about endospores and how an endospore is actually formed. So we're going to talk about the process of sporulation and how this process of sporulation can impact certain infections. Okay, so let's start with the biofilms. Okay, we talked about this when we talked about the bacterial growth curve. So this happens in the late log phase. Okay, so we're about done with the log phase. We're almost to the top of that plateau. Well, the thing is, with a biofilm, we have to have a large amount of bacteria in order to form one, which is why we don't see it happening until late log phase. Late log phase is when we can get quorum sensing. Okay, so we define a biofilm as a mass of bacteria that stick to and multiply on a solid surface. Now, that solid surface can be anything from teeth. If you have gone to get your teeth cleaned in the last year, you have had a biofilm scraped off your teeth because plaque is a biofilm. Now, hopefully, your teeth don't look like this person's, with the receding gums and the really yellow teeth. Like you can literally see the biofilm on this guy. Guys, mouths are gross. So gross. Okay, so it could be a solid surface like teeth. It could be a hospital surface. It could be a catheter. Um, in fact, this is where we see a lot of biofilms forming in the medical field, is because hospital catheters aren't um, kept as sterile as they could when they were being inserted, which is going to cause some problems with the patient. So with our biofilms, they can be a single species biofilm or they can be multiple species biofilms. Now, things like your plaque, there's a lot of bacteria in your mouth, which is also why it's gross. So the plaque in your mouth is going to be a multi-species biofilm. Okay, and the thing is, whether it's a single species or or multiple species, they have to be able to talk to each other. Okay, they have to be able to communicate. So that's going to happen with the quorum sensing. But one of the things that happens with biofilms, in order for the biofilm to be made, um, each of the bacterial cells need to start to make what's called EPS, okay, extracellular polymeric substances. Okay, so this is going to make a sticky matrix, a sticky substance that is going to surround the bacteria, and it's going to help them stick to this surface, whether it be an organic or inorganic surface. Okay, so let's look at this guy's teeth. It might be a lady, this person's teeth. So if we look at the electron micrograph, the brown spots, this is the extracellular polymeric substance. This is the EPS layer. Okay, the bacteria are literally going to be stuck in it. This is what's going to allow them to stick to that surface. But the thing is, once the biofilm is formed and we have this EPS layer, this makes it good for the bacteria, bad for us. So it's going to act like a protective shield, and it's going to make this biofilm is going to now be resistant to chemicals and antibiotics, meaning antibiotics can't get in and kill the bacteria in here. So this is one of the reasons why that you can go on an antibiotics course and still have plaque afterwards, because that biofilm, that plaque, is actually protecting those bacteria from not only the antibiotic, but from your immune system. So it's a pretty safe place for them to live. Okay, which brings us to how biofilms form. So we're going to go through this really quickly, and then we'll watch the animation for it from the book. So with biofilms really need quorum sensing to happen in order to coordinate actions between the different bacterial cells and for them to communicate. Okay, so this EPS layer and this formation of the biofilm is going to make this biofilm a very safe place for the bacteria because it's going to be very hard to destroy. Okay, so all of our biofilms are going to start with what we call planktonic cells. Okay, so these are just going to be single microbes in the environment. So they're going to kind of swim around like microbes do, sensing their environment until they find a good one. Okay, so what they're going to do is they're going to find a good environment and they're going to find a surface. They're going to kind of stick to it. They're going to use their flagella in order to do this. And once they have their flagella stuck to that surface, they're going to form what is called a monolayer. That adhesion of the flagella to that surface is going to kick off a different chemical pathway. They're going to start excreting this EPS layer, the sticky substance, which is going to allow them to stick to that surface. And at this point in the process, they are going to dissolve their flagella, so they're going to get rid of it because they're now stuck in this substance, and they're going to have what is called this twitching motility. So they're going to go back and forth until they kind of form this microcolony. We'll see this more in the. I'm not very good at explaining this. We'll see more of this in the animation. Okay, so we have this sticky substance. We're stuck in it. We're twitching. We're going to start to excrete more of that EPS layer. So it's going to start to build up and it's going to start to cover the cells. Okay, and then we're going to get these cells started to go through cell division, so we're going to get more and more, and we're going to literally form these fingers of biofilm, these biofilm towers that are going to have a lot of bacteria in it. And the bacteria are going to stay in these towers in this biofilm for as long as the conditions are good. Now, these towers might get a little overcrowded, the environment change, it might go from a good environment to a bad environment, nutrients might be scarce. So what can happen is these cells are communicating in this biolayer, and as the biolayer builds up and the communication molecules build up, that's when they're going to start to quorum sense. Okay, so when environments get bad, these cells can make a substance that are going to dissolve the biofilm. They're going to remake their flagella, and they're going to swim out of the biofilm and go towards greener pastures. So this whole cycle is going to start over again. So let's watch the animation, which I think is going to do probably a better job than Miss Harker did explaining how this whole thing happens.
Bacteria were once thought of as unicellular organisms incapable of differentiation or communication. but we now know that many, if not most, bacteria form specialized surface-attached communities called biofilms. Indeed, within aquatic environments, bacteria are found mainly associated with surfaces. This biofilm is a plaque that forms on the surface of a tooth. Plaque is a mixed species biofilm, but single species biofilms also form in nature. For example, single species biofilms of Pseudomonas aeruginosa can form on the lungs of cystic fibrosis patients. The first stage of biofilm formation occurs when a specific environmental signal triggers unattached planktonic cells to attach to nearby inanimate surfaces by means of flagella, pili, lipopolysaccharides, or other cell surface appendages. The attached cells begin to coat that surface with an organic monolayer of polysaccharides or glycoproteins to which more planktonic cells can attach. As the cells enter into biofilm formation, they no longer maintain their flagella and instead may move along surfaces using a twitching motility that involves the extension and retraction of a specific type of pilus. Ultimately, they stop moving and firmly attach to the surface. As more and more cells bind to the surface and divide, they form microcolonies and can begin to communicate with each other by sending and receiving chemical signals in a process called quorum sensing. These chemical signals are continually made and secreted by individual cells. Once a population reaches a certain number, analogous to an organizational quorum, the chemical signal reaches a specific concentration that the cells can sense. This concentration triggers genetically regulated changes that cause cells to bind tenaciously to the substrate and to each other. Quorum sensing may influence the amount of extracellular matrix produced by the cells. This thick, slimy material consists of polysaccharide polymers, called exopolysaccharides, and entrapped organic and inorganic materials. These exopolysaccharides increase the antibiotic resistance of residents within the biofilm by limiting antibiotic access to the biofilm's center. The chemical signal molecules also stimulate production of some antibiotic resistance mechanisms. As the biofilm matures, the amalgam of adherent bacteria and matrix forms columns and streamers, creating channels through which nutrients flow. Although most of the bacteria adhere to the biofilm, some planktonic cells leave for new environments. Here is an image of the lumpy, uneven biofilms created by a mucoid environmental strain of P. aeruginosa. Biofilms allow bacteria to stay where food is plentiful. Why should a microbe travel off to hunt for food when it's already available? Once nutrients become scarce, however, individuals will detach from the community to forage for new sources of nutrients. P. aeruginosa produces an enzyme that can strip away the exopolysaccharides and will resynthesize flagella. Cells leaving the biofilm revert to their flagellated planktonic forms and can swim to new locations where nutrients are more plentiful. A hallmark of infectious diseases in which biofilms play a prominent role is their chronic nature. A biofilm infection may linger for months, years, or even a lifetime.
Okay, I really like that animation. So there's a lot of infections where biofilms are created. So we've talked about catheters and medical field. This is a common source of sepsis in a patient is if they have a catheter that builds a biofilm, because it's hard to get rid of. Um, so with cystic fibrosis patients, they never really get rid of that Pseudomonas aeruginosa. So one of the things that happens with cystic fibrosis patients is they often have to wear a special vest or jacket that's going to kind of pulsate. So it's going to help those cystic fibrosis patients break up those mucus layers so that biofilm can't form as well. Because if that biofilm forms, they're going to come down with pneumonia. And a lot of people with cystic fibrosis die from that pneumonia. Back in the day before we had these special vests and jackets, what they had to do is a couple times of the day, they literally had to have their backs pounded to break up all the mucus layers in their lungs so they could prevent that buildup. But there's a lot of infections that use biofilms in order to stay relevant. They're immune resistant, they're antibiotic resistant, so you can't really get rid of that infection once you have it. Okay, so that's biofilms. They will pop up a few times between now and the end of the semester again. Um, as I said, they are a current problem in the medical community. So one of the things that we need to talk about, the last thing we're going to talk about actually, are endospores and how they form. So one of the things about endospores is they're really only formed by gram-positive bacteria. So these gram-positives are going to be things like bacillus species, like Bacillus anthracis, which is really bad for bioterrorism. A lot of you are probably too young to remember the bioterrorist attacks with anthrax after 9/11 happened. One of the problems was they used the endospores for Bacillus anthracis, so it was dormant until it got in the air and started to infect people. Okay, so when bacteria that forms endospores are in their dormant form, this is when they're in their endospore form, they don't require nutrition or energy, they're just there. So things like Clostridium species, like Clostridium botulinum and Clostridium tetani, these can cause disease where you wouldn't originally think it would be. So like Clostridium botulinum, we saw in a case study earlier that was in contaminated canned food. So with Clostridium tetani, this causes tetanus. So if you go and step on an old rusty nail, one of the reasons why you get a tetanus shot is because Clostridium tetani can form these endospores that can survive on that nail for a long time. And so when that nail goes through your foot and transfers the endospores into the inside of you through the puncture, they now have a good environment where they're going to become not dormant, and they're going to resuscitate themselves and they can grow and divide and cause an infection. The thing with endospores is they can last decades up in centuries before they have to be reactivated. So they can stay dormant for a really long time. Okay, so endospores are highly resistant to destruction. They can resist being dried out. They can resist heat. So like if we boil endospores, we can't get it hot enough to destroy the endospore. They're antibiotic resistant, they're chemical resistant. In fact, the only ways that we really can get rid of endospores reliably is to use high energy radiation like X-rays and gamma rays. In fact, a lot of food nowadays, before it goes through the process of canning or to be delivered to grocery stores, is actually irradiated. So with X-rays, before it leaves to hit the shelves of the market or to be canned so that we can kill those endospores. Also autoclaving. So we're going to use those high pressure conditions to drive the temperature of water above boiling, so to about 121 degrees Celsius, so that we can kill that endospore. Those are really the only two reliable ways to get rid of endospores. In fact, endospores have lasted millennia. In fact, we talked about previously the fact that scientists found endospores of a bacteria in amber. This is actually the picture from it. So there's this bee that got stuck in amber back in the day. And what they noticed while they're studying it is that underneath the microscope, they found that there was these spores on the outside of it and in the bee's gut. So they carefully harvested those endospores and they germinated them, so they grew them in the lab. So I got this from Dr. Burroughs. It's like Paleogene Park, where we reanimated something that's millions of years old. So this link down here is actually to the article where they published their findings from reanimating this bacteria. Scientists do some weird but cool things. Okay, so let's talk about the sporulation process. So let's talk about vegetative growth. So vegetative growth, even though it doesn't sound like it, vegetative growth is just normal growth. So this is how the cell grows, copies its DNA, and divides into daughter cells. Okay, so this is the normal cell process right here. Okay, so if the cell is in good conditions, everything's going fine, it's going to just go through multiple rounds of vegetative growth and make more and more cells. Now, if conditions change, it can flip the switch and go from this vegetative growth stage to this sporulation stage. Okay, so the first step is the same. We are going to copy the genome. So we're going to have two circular chromosomes. So what's going to happen So here, our septation is going to happen in the middle. So in sporulation, we're going to have the septum that is going to form closer to one side than the other. We're going to pump that DNA into that. They're going to pump it past the septum. So we're going to have two genomes on two distinct sides of the bacteria. Okay, so the septum is going to form, and this is going to form what is called the forespore, and the rest of it's going to be the mother cell, so what's remaining of the living cell. Okay, so the forespore is going to be engulfed by the mother cell. This is going to give it a second membrane around it. Okay, so once it has the second membrane, it's going to start to make a special peptidoglycan layer that's going to be formed and pumped into the space between the two membranes. This is going to form what is called the cortex. So at this point, the mother cell's genome is going to start to degrade. It's going to start to break down. So the mother cell is going to start to die. So after the cortex is formed, it's going to go through this process where it's going to start to make the spore coat. Okay, this is going to be the coating that's going to make it both antibiotic, heat, and chemical resistant. So what's going to happen is it's going to make this spore coat, and it's going to use dipicolinic acid and calcium in the spore coat in order to make it resistant. So it's going to kind of dry it out so that it's going to be dormant. Okay, so the mother cell is going to officially die. It's going to go through apoptosis. It's going to release the endospore. The endospore is going to hang out in the environment until conditions change or it hitches a ride somewhere else. So once conditions change, it can go through a process called germination where it kind of reactivates. So it starts to break down the spore coat and goes back to its vegetative growth form. So it becomes a parent cell again and then goes through cell division as it did before the whole sporulation process. So the last thing we're going to do is we're going to watch an animation about this because this is something that bears repeating because this is not a simple process. I would know vaguely how this happens. You don't need to memorize what happens at every step. So let's watch that video.
When starved for nutrients, certain gram-positive genera, including some important pathogens, can develop dormant heat and desiccation-resistant spores. Most of our knowledge of bacterial sporulation comes from the gram-positive soil bacterium Bacillus subtilis. When growing in rich media, this microbe undergoes normal vegetative growth. B. subtilis can replicate every 30 to 60 minutes. In contrast, starvation initiates sporulation, an elaborate 8-hour genetic program that ultimately yields a spore. In the first stage of sporulation, the septum forms near one pole of the cell rather than in the middle. The DNA replicates and extends into an axial filament that spans the cell's length. During the next stage, the septum separates the forespore, which will ultimately become the spore, from the larger mother cell. DNA is pumped through the septum until each compartment gets a chromosome. The mother cell now engulfs the forespore, surrounding it with a second membrane. The mother cell chromosome is eventually destroyed. A cortex, made up of a thick peptidoglycan layer, is placed between the two membranes surrounding the forespore. This cortex will be important in providing heat resistance and maintaining dormancy to the spore. Layers of coat proteins are deposited on the outer membrane. In the next stage, dipicolinic acid and calcium are incorporated into the spore. Dipicolinic acid is important in maintaining dormancy and in promoting spore dehydration, a characteristic that makes a spore heat resistant. The mother cell, now called a sporangium, dies and releases the mature spore. Spores are resistant to many environmental stresses that would kill vegetative cells. The nature of this resistance is due in part to the spores having only 10 to 30% of a vegetative cell's water content. But spores also contain small, acid-soluble proteins that bind to and protect DNA from damage by ultraviolet light and various toxic chemicals. Fully mature spores can exist in soil for at least 50 to 100 years and have been known to last thousands and even millions of years. Once proper nutrient conditions arise, Another genetic program, called germination, is triggered to wake the dormant cell, dissolve the spore coat, and release a viable vegetative cell.
Okay, I really like that video. It kind of goes through things I forgot, like the fact that it has considerably less water than a normal cell, which is going to make it heat resistant. So that is chapter 6.6. We are done with chapter 6. We now know how biofilms form. We know how endospores develop. I would know basically what this does, know when quorum sensing happens. So that's it for 6.6. Let's do one more review question before we head on to chapter 7. Okay, so our final review question says bacterial organisms form a biofilm to A, claim territory and fight off competing organisms of a different species. B, attach to a surface in an area where food is plentiful. C, find a food source where nutrients are scarce. Or D, mate with bacterial organisms of the same or different species. Okay, so pause the video, flip through your notes, and see if you can find the answer. Okay, the answer for this question is B. It's going to allow it to attach to a surface in an area where food is plentiful. Okay, so there's one of these that we can automatically just cross out because it's not even true, and that would be D. So bacteria go through binary fission, which is a form of asexual reproduction. They don't need other bacterial organisms in order to mate or make little bacterias. So D is wrong. Okay, A could potentially be right, but biofilm is not going to allow it to fight with other species. So that really leaves B and C. And now we know that biofilms are for good environments, so this is where food's going to be plentiful, which is why B is the answer. Okay, so that is chapter 6.6. Your homework is going to be due soon. Look on Canvas to know when that is. So we will see you in the video for 7.1.